Hi, my name is Michelle Sostner, and I studied the effect of UK5099 on HT22 cell protection from ferroptosis. Ferroptosis is a form of programmed cell death, and it is seen in numerous neurological diseases such as Alzheimer's. This form of cell death can be triggered through the inhibition of the Xc- transporter. The Xc- transporter is an antiporter, which means that it can import and export different amino acids. In this case, it imports cysteine and exports glutamate. This transporter can also be inhibited by large concentrations of glutamate, and heavy exposure of glutamate can cause ATP depletion, and the inhibition of the Xc- transporter can also cause glutathione depletion. And these are all things that can cause cell death. In this experiment, I'll be using Erastin, and this is a drug that induces ferroptosis. I will also be using HT22 cells. These are mouse hippocampal cells. They are immortalized, which means that they can divide indefinitely. And these types of cells are commonly used in glutamate excitotoxicity studies because they express the Xc- transporter. These cells are also undifferentiated neuroblasts, which means that they are all the same. I will also be using the drug UK5099, which is a drug that's known to inhibit the mitochondrial pyruvate carrier, also known as the MPC. Through MPC inhibition, the TCA cycle will utilize the intracellular glutamate, and that will result in less glutamate being in the synapse. Blocking pyruvate will also mean that the mitochondria needs to find another way to use energy, and it uses the intracellular glutamate. So the UK5099 drug can essentially protect neuronal communication and cell death. Previous studies have used the UK5099 drug in cortical neurons, and the results were amazing, and it showed that the neurons were very well protected, with the survival rate being at almost 100%. However, the MPC inhibitor UK5099 has not yet been tested in the HT22 cells. So I asked the question, can ferroptosis be inhibited in HT22 cells by UK5099, an MPC inhibitor? To answer this question, I hypothesized that by inhibiting the MPC, the UK5099 will protect the HT22 cells from ferroptosis. To begin, we had to determine the LD50. This is where around 50% of the cells will die in a certain cell density. So different cell densities were placed, ranging from 30,000 to 310,000 cells per ml. And in the wells with the Erastin, there are 49.5 microliters of HT22 cells with their media, and then half a microliter of Erastin. And in the wells without the Erastin, there will be the same amount of HT22 cell with media, and then just one more half a microliter of the media. So to determine the LD50, we did an MTTSA. So after we let the Erastin sit in the HT22 cells overnight, we conducted the MTTSA the next morning. MTT is a yellow non-toxic dye, and it monitors cell viability. The yellow MTT will be converted into purple formazan crystals. And the more purple a well looks, or the more crystals there are in a well, it means there's more cell viability. And then after a 45-minute prescribed incubation period, this plate is put into a spectrophotometer, which is a machine that just quantifies the data. In addition to an MTTSA, we did a live datassay to visually confirm the data that we received from our MTT. So I used calcine AM, which will dye the alive cells to a fluorescent green, and we also used ethidium homodimer, which will dye the dead cells to a fluorescent red. And through both of these assays, the LD50 was determined to be 180,000 cells per ml. This LD50 determination process was completed twice to ensure that these findings were reproducible, which they were. After we determined the LD50, this 180,000 cell per ml density was then tested for UK5099 protection. So these cells were put under different conditions. We had our negative control, and then we had our second row, which was NAC, which is a positive control. And then in columns 3 through 6, we used UK5099 at 2 different stocks, and as well as different amounts. And all of these distinct conditions were used so that we could rule out any possible confounders as to how much or how the UK5099 could help these HT22 cells. So just like in the LD50, we used an MTTSA and a live data assay, and these are the results from the MTTSA. So as you can see, the control, around 50% of cells died. In the NAC, the positive control, almost all the cells survived, which was expected. But then when we get to the UK5099 cells, it seems as though the cells were not protected. And it seems around 50% of cells still died. And even adding more of the UK5099, we noticed that it did not really help, which was very surprising based on such positive results in the previous studies. And to visually confirm this data, we did a live assay. And as you can see, the control, it looks about 50% of the cells died. And then in the UKs, the cell with the Erastin looks around the same as the control did, which again was very surprising. And then in the NAC, it was the positive control and almost all cells survived, which was to be expected. So this experiment was just an assessment of whether the mitochondrial pyruvate transport inhibition could potentially protect against ferroptosis in H222 cells, just like it can in cortical neurons. But we found that the UK actually had no effect on ferroptosis. But this lack of effect cannot be attributed to the toxicity of the drug because it did not exacerbate cell death. It just didn't necessarily help in protecting more cells. And based on the results of the experiment, we came up with a few limitations on the experimental design that could also explain such negative results. So we didn't have an independent measure of MPC inhibition or an independent measure of glutamate in the cells. And the UK could have also been unstable in the media that was used. Something could have gotten into our drug solution, and that could have hindered its effects on the H22 cells. Another main difference between this experiment and the previous studies experiments was that the previous positive results were obtained in primary cultured neurons. So these neurons are differentiated, they do not divide, and they do have synaptic connections, whereas the HD22 cells are immature neuroblasts, and they are not differentiated, they do divide, and they do not have synaptic connections. So in conclusion, the results did not affirm the hypothesis, and because of that, we came up with a few different limitations and errors that could have been present in the duration of the experiment. In the future, I'd like to conduct an experiment that could directly address the majority of these limitations or the errors that were possibly made. So for example, I would like to complete the same exact experiment with both the H22 cells and cortical neurons at the same time and then directly compare the data. And I would also like to do more research on the mitochondria's role in ferroptosis to try and understand why there's such a drastic difference in results in different types of cells. And that research could hopefully close a gap as to what the mitochondria does in ferroptosis and how that could directly affect the results of the experiment. I would like to thank my science research mentor as well as my science research teacher, and these are my sources. Thank you very much.